Today's PowerPoint is about carbohydrates. So we're following up from our introduction to organic chemistry and functional groups and the idea of polymerization from monomer units. And before starting the PowerPoint, you may want to watch this video that I found on YouTube. It gives some great information about carbs and a lot of the material that's going to be in this PowerPoint. So if you have time, do watch. So in terms of definition, carbohydrates are organic molecules or biomolecules that contain hydroxyl groups and either one aldehyde or ketone in the linear form. And in general, the molecular formula is, as you can see on this slide here, CH2O, and N would be the number of carbon, for example. So if we were talking about some of the sugars we were looking at when we were talking about isomers, glucose, fructose, and galactose, the N number there would be 6. So we would have our C6H12O6. And that follows from that we would have a 1 to 2 to 1 ratio between carbon, hydrogen, and oxygen. The monomer unit of our sugars are what we call monosaccharides. And in order to make our long chain sugars that we use for energy storage and for structural reasons, we would take our monomer units and we would join them together to form a long chain of sugars, which we would call a polysaccharide. So our most common monomer units that certainly the ones that we're going to be focused on in this biology course will be the pentoses, which are 5 carbon sugars. The pentoses have 5 functional groups, so 1 per carbon, and that includes 4 OH or hydroxyl groups and 1 aldehyde or ketone, which again we can see much more easily when the sugar is in linear form. In this case, we're seeing our 2 most important pentose sugars, the 2 that we're going to be focused on for our purposes, not only in our biochemistry unit, but also in our genetics unit, and that is ribose and deoxyribose that we can see pictured here. And I'm sure you're familiar with those names. You know that that's the sugar that's contained in DNA and RNA. And if you have a look, they are identical except for the bottom right hand OH group and H group. So if you look at ribose sugar in the, I guess if you like, bottom right hand corner, you can see that there is an OH group attached to that carbon. And if you look at the deoxyribose, you can see it's just a hydrogen. So deoxy meaning one less oxygen. We'll also spend a lot of time looking at the hexoses, which are 6 carbon sugars, which we've already spent considerable time looking at when we were looking at isomers. 6 carbon sugars have 6 functional groups, which include 5 OH groups and 1 aldehyde or ketone, most easily seen when they are in linear form. And there's our examples that we looked at before: glucose, galactose, and fructose, recalling that galactose and glucose are stereoisomers, and glucose and fructose are structural isomers. Both pentoses and hexoses will form ring structures in water when the polarity in the water causes some of our functional groups to interact with each other. So when we actually see a lot of the drawings of our monosaccharides, they will be in ring structures, as we saw in the previous slide. However, when they're in their linear form, like the glucose and fructose on the right-hand side of the slide, we can more easily see their aldehyde and ketone groups. So if you look at glucose here, you can see that glucose is an aldehyde. It's got the carbonyl group on the terminal carbon. And you can see that fructose is a ketone sugar, and the carbonyl group is in one of the internal carbons. And in the diagrams at the bottom here, you can see how linear fructose is going to sort of bend in the water when it's in liquid water. And what happens is the carbon, in this case, carbon number 2, is going to associate with carbon number 5, and we see a little bit of really a redox reaction where we've got some changing of electrons, and we get a little bit of a rearrangement to form the ring structure. And something similar is going to happen with glucose, except it's carbon number 1 to carbon number 5. And because the diagram is in color, you can maybe see a little bit more clearly where the carbons and hydrogens and so on goes. We have a breaking of a double bond and the formation of a new single bond to form our ring-structured carbon. So glucose is definitely our most important carbohydrate monomer unit. It's going to be very important in our second unit when we talk about cellular respiration and photosynthesis. So we're going to look at how glucose is made and how it's broken down in nature. And we're going to see why, a little bit at least, why it makes a good energy storage molecule, and how it's nice and convenient because it's easy to form the long chain for storage, and it's also easy to break up that chain and use the glucose for whatever it is that we need in terms of energy requirements. It's also a sort of a transitional molecule in the sense that we can make some small changes and use components of glucose to synthesize fatty acids, as a for instance, or a glycerol molecule that we use in lipids as well, which we'll see coming up, and can also be used to create amino acids for the formation of protein. And some of it can also be used to give us the components of nucleic acids for DNA and RNA production. So a very important molecule for all kinds of different reasons. If we take two of our monomer units and we put them together, we form something called a disaccharide. And only certain sugars will stay sort of stable as a disaccharide or functionally hang around as a disaccharide. So what happens is that what we call a glycosidic linkage is formed between the two monosaccharides. This reaction will be what we referred to earlier as a one of those reactions where we're removing the water molecule. So it's a condensation reaction or a dehydration synthesis. So water is being taken away. For most of the disaccharides that form, what will happen is that the carbon number 1 of one of the monosaccharides will join or form a bond with carbon number 4 or carbon number 6 of the other monosaccharide unit. Depending on whether we're seeing sort of straight lines or branches. There is one exception in the disaccharides that we're going to look at today, and that is between glucose and fructose, and we'll see that that's a little bit of a different setup just because of the way the carbons are numbered. So here are the 3 disaccharides that I expect you guys to be able to remember. And keep in mind that the last one that's there, maltose, I'll expect you to be able to draw because glucose is the one sugar that I'm going to require that you know how to draw the structure for. So 2 glucose molecules together are something that I would expect that you would have no trouble drawing once we're finished with our carbohydrates. Our first disaccharide in the table here is called sucrose, and that's our table sugar. We're pretty familiar with that. It is a disaccharide formed between glucose and fructose, and it forms this odd link that I mentioned where we get a 1,2 link, an alpha 1,2, alpha sort of designating the kind of sugars that we can digest. So alpha 1,2 linkage that we'll look at in a graphic in just a minute. And The second sugar that we have, disaccharide, that we have in this table is lactose. And I'm sure you've heard of that sugar before because lots of people are lactose intolerant, which means they can't actually break down this sugar. And in fact, mammals generally lose the ability to create the enzyme to break down this particular sugar after their age of, when they're usually weaned from their mother's milk. And humans are very unusual in the sense that we can continue to digest this sugar. So in fact, the ability to digest lactose was actually a mutation. And so I guess the people that can drink milk are mutants, and the people who cannot drink milk are more the norm for mammals. And the final sugar on the table here is maltose, which is malt sugar and often used for fermentation of alcoholic beverages such as beer. Both lactose and maltose are formed from 1,4 bonds, and we'll have a look at what that looks like. So in this graphic, we see that 1,2 glycosidic linkage, and don't forget the name glycosidic linkage or glycosidic bond. It does often come up in multiple choice and on tests and so on. So here we've got our glucose in sort of an orangey color, and we can see carbon number 1 of glucose is forming a bond, a glycosidic linkage, 1,2 glycosidic linkage with the carbon number 2 of the fructose molecule here to form sucrose. Then we've got our galactose and glucose, where we've got carbon number 1 of galactose to carbon number 4 of glucose, which is giving us our lactose sugar. And the last one that's there, we've got 2 glucose molecules that are being bonded together, carbon 1 to carbon 4. So if we have the right circumstances, we can, as living biological systems, polymerize the monosaccharide units into much longer structures, usually for formation of a particular kind of material, say like cellulose, not that we make them, but that plants can make. So some kind of structural chain, or for storage for later use. So we put a little bit of glucose aside for later use. For instance, in mammals, in a lot of animals, we will store a little bit of glucose as glycogen, which is an easily accessible material that we can quickly convert if we need to. So we often will store that in our muscles and in our liver, just in case a saber-toothed tiger tries to eat us, and we can run away very quickly. So if you had an adrenaline surge or epinephrine, you very quickly break this glucose down from the glycogen long chain polymer into glucose and be able to use that as a fuel to get the heck out of the way. So polysaccharides are very important both for energy and for structure. So polysaccharides are formed usually primarily from glucose molecules, and what happens is we get a lot of 1,4 linkages, but we also get a whole lot of the 1,6 linkages as little side chains, and we'll have a look at what that structure looks like. So if we, depending on what the material is, you may find that it only has 1,4 links, and we'll see that that's the case with one of the more simple kind of plant starches. With the more complex plant starches, you're going to get more branching, and with the material I was just mentioning, glycogen, you're going to get a lot of branching, and you can find, depending on how much we're storing, that you can get hundreds to thousands of monosaccharides in these long polysaccharide chains. The more branching that we have, the less solubility we have. So I'm sure you guys have all experienced what happens when you mix cornstarch with water. Cornstarch has quite a lot of branching in it, which makes it a little bit difficult to dissolve in water. And here's a little look at the examples. So here in this little graphic, there's a look at glycogen, which has, you can see, sort of 1,4 bonds to form the long strings, if you like, and then we've got branches off 1,6, and we'll again have a closer look at that. And there's cellulose, which is not a branch material. You can see that it's got long chains that give sort of fibrous structural kinds of qualities to cellulose, which give us the long sort of stringy feeling that we get from the cell walls of a plant. And over on the side, we see starch here. Now, this is a very simple starch that we're seeing here, no branching, but there's also a more complex starch that has some branching to it, and we'll have a look at that too. Okay, so if we're going to categorize our polysaccharides, as I mentioned earlier, we can do that in sort of two big categories. The first one here being storage polysaccharides, and we're going to look at the plant storage polysaccharides. So by storage, what I mean is that we are storing the glucose molecules away so that we can use them for energy purposes or for some short-term needs, maybe for construction of other biomolecules. So we're not using them to make part of a cell, for instance. We're hanging on to it so that we can carry out some biochemical reactions. So the most simple kind of storage polysaccharide is a plant starch, we call it, called amylose. And that is a simple structure that is a long chain with just 1,4 glucose glycosidic linkages between the glucose molecules. In plants, we also get a more complex starch, which we call amylopectin. Of course, there's lots of subheadings under these. These are sort of larger categories, but amylopectin is a very long chain storage polysaccharide in plants that not only has the 1,4 glycosidic linkages, but also has 1,6 alpha 1,6 glycosidic linkages. And those 1,6 links usually occur approximately every 20 or 25 glucose molecules. And this is a little bit blurry, but hopefully you can see the amylose is the top part of the graphic, and it's very linear, straight line, glucose to glucose, just like we were looking at before, the 1,4 linkage that we were seeing earlier. And the bottom part of this graphic is amylopectin. And again, you can see sort of two chains of long 1,4 linked glucose, but then partway through the chain you can see a branch that's coming off between carbon number 1 of the sort of bottom chain, carbon number 6 of the bottom chain, and attaching to carbon number 1 of the top chain, and that is our 1,6 branch. So this is plant starch. In animals, we make that material I was mentioning earlier, not animal starch. We don't call it that, but it's basically the same thing. We call it glycogen. Glycogen is very much like amylopectin, the more complex plant starch, but the branching occurs much more regularly. Between every, say, 6 or 10 glucose molecules, you're going to have a branch. And as mentioned earlier, it is going to be stored in liver and muscle for easy access. It's a little more water soluble than plant starch, so you don't quite get that. Not that you're going to take that from people and sort of try to mix it with water, but if you did, it would be a little bit easier to mix than cornstarch, as an example. In the glycogen graphic that's here, so in this graphic at the bottom here, we can see the glycogen molecule looks very much like amylopectin, same 1,6 branching that we can see, and perhaps a little clearer than what we could see with the amylopectin diagram. The second large category of long chain monomer units for the carbohydrates, polysaccharides, are structural polysaccharides. And we're going to look at two examples, really. Cellulose is, in fact, the major component in cell walls of plants, and is one of the most abundant materials on the planet. It is made of a beta glucose, and we did talk about a little bit earlier when we were talking about, I can't remember what, something. We were talking about the fact that alpha glucose is a form of, I guess when we were talking about isomers, is a form of glucose that we can digest rather easily, where beta glucose is a form that we do not. So in terms of biological activity, alpha glucose works pretty well for us, and that's why it makes a good energy molecule. So our storage polysaccharides, the ones we were talking about just a few minutes ago, are made from alpha glucose. The structural polysaccharides made from beta glucose, so we don't digest them in the same way, and they make good structural components for cells. So the beta glucose links together in much the same way. You get, instead of alpha 1,4 linkages, beta 1,4 linkages. And because of that, we don't have a digestive enzyme that will fit this link and break it down, so we cannot digest it. So it ends up being good fiber or roughage for us. And we can see an example of what that might look like. And we saw in a previous slide that it ends up giving quite a linear structure, which makes sense. If you've ever eaten celery, all those nice sort of long fibrous sections that you can they crunch through with the celery, you're mostly chewing on cellulose. So we don't really have the same, the equivalent of a nice general structural polysaccharide like cellulose in the animal world, but chitin is a pretty nice example. Chitin is actually modified glucose, not a pure glucose molecule, and what that modification is a nitrogen group and amine group. We find chitin in insect skeletons and in the cell walls of fungus. And if you've ever stepped on a great big cockroach and heard that crunch, then what you're hearing is, for the most part, chitin. So the modified glucose has an amine group on it, as I was mentioning, and you can see here at the graphic, if you have a look, it looks very much like our glucose molecule, except on carbon number 2, instead of having an OH group, we have an NH2 or an amine group in its place.